Good afternoon. I'm Igor David, and together with Win Arias, I am co-host of today's speaker. And as Dr. Collins had to cancel recently, well, these microphones, of course, are not friendly to me, but they are going to be much more friendly to Didier, who is going to speak to you much longer than I am, hopefully. So Didier Stanier is a leader in the area of cardiovascular and endodermal organ development. And he works with zebrafish, as the title says, or indicates. So after his education in Belgium and the UK, he came to this country, obtained a PhD at Harvard, and then he joined the large screen of zebrafish mutants at the Mass General and was involved in a leading way in isolating many developmental mutants in this organism that have really started the entire field. Then moving to UCSF, he quickly became a leader in the field, and he is now professor of biochemistry and biophysics, the co-director of the developmental and stem cell biology program, associate director of the liver center, and involved in various other efforts that I won't take the time to mention. So among the major insights that emerged from his work are the importance of endoderm in heart development. That came about by finding that several mutants that affect the heart turned out to be endodermal genes that are expressed and important for endoderm development. For example, Casanova, which is a SOX gene, Faust, which encodes GATA5, Bonnie and Clyde, which is a mixed family homeodomain factor. All of these are involved in endoderm development, but critical as well in heart development. In addition, he studied a gene called miles apart, which turned out to encode a sphingosine 1-phosphate receptor, which is required for cells to migrate to the midline to form the heart. And this work again introduced a totally novel direction and pathway into the field. But he did not just take endoderm development as a stepping stone to studying the heart, which it was, but in addition, he went on to study endoderm organ development in great detail, provided the most explicit and detailed characterization of the development and insights into the molecular mechanisms of endoderm development in fish, including such findings as the shaping of the liver by the vasculature, relationships between pancreas and liver, and so forth. So obviously these various kinds of studies lend themselves to applications, so studying the relationship to possible medical applications, and that's what he will talk about today. So the title of his lecture, which went off the screen, is Curing Diabetes One Fish at a Time, the Long Road of Translational Research. And I asked Didier Stanier to give his talk.
Thank you very much for the very nice introduction. Can you all hear me? And it's a great honor and pleasure to be here. And I want to thank again Dr. David and Arias for the invitation. And as Dr. David indicated, what I'm going to be talking about today is using the fish for more medically relevant purposes, that is, using it to screen for small molecules that might be relevant for diabetes. But before I do that, I will tell you about the biology of pancreas development and also how the fish system might be used for other approaches, non-small molecule approaches to curing diabetes. So as Dr. Dawood indicated, my lab is interested in vertebrate organ development and function, and we've been interested in developmental issues such as those listed here, but also in organ function and homeostasis. And the general approach, as was mentioned, has been one of forward genetics, that is to mutagenize the genome and then look for interesting phenotypes in these various organ systems, the cardiovascular system, as well as the liver and pancreas. And as was mentioned, we came into the liver and pancreas from studying a specific subset of heart mutants. So, and to carry out this forward genetic approach, we've been using the zebrafish along with now about 700 other labs around the world, including about 15 labs here at the NIH. And the reason that the field of vertebrate development and vertebrate biology has embraced the zebrafish are several, and some of them are shown on this slide. And probably the rapid development and transparency of the fish embryo, including, of course, the access of the embryo from an early stage due to the external fertilization, has made this a very malleable system. And this, along with the forward genetics that was mentioned, as well as the fact that the organs are of relatively small size, make it, again, a very attractive system to try and understand developmental processes at the single cell level. And I will show you some of the examples along these lines. So if you think about the zebrafish, and there are really two main advantages to using it in the system. And one of them is the fact that one can do live imaging because of the external development and the transparency in the embryo, and also the ease with which one can make transgenic fish. And so, for example, this is a SPIM imaging of the 7-day-old heart. And SPIM, of course, as you know, stands for selective plane illumination microscopy. So what you're looking at here is a line where GFP is expressed in the endothelial cells of the heart, the endocardial cells, and DS red in the blood. You can see the atrial chamber, the ventricular chamber, the bulbous arteriosus. You see the valve flaps between the atrium and the ventricle. And I think, again, being able to look at some of these processes or some of these organs as they function in real time has allowed us to, and others in the field, to ask very specific questions about organ formation and function. And closer to what I'll be talking about today, we're looking at here early endodermal cells. These are SOX17 expressing endodermal cells. We're looking at eutrophin GFP transgene here in this region of the embryo. These single endodermal cells are at this point migrating fairly randomly, and then will migrate towards the dorsal axis, where they will form the endodermal sheet from where the endodermal organs will form. And besides the imaging, the ability to collect large number of animals has made it possible then to do large-scale screens, not only the forward genetic screens that you heard about, initially carried out at MGH and also in Tübingen in Germany, but also more recently chemical screens. So small molecule screens targeted specific processes. And so, for example, what I'll be telling you about today is one screen where we're looking for small molecule enhancers of beta cell regeneration, and another screen where we've been looking for regulators of gluconeogenesis. So again, trying to attack glucose or restore glucose homeostasis from two different angles. So just to say a few words about forward genetics, the initial screens were done essentially looking for embryonic lethals. And as the field has become more sophisticated, we and others, again, have taken advantage of transgenesis and to look, for example, at genes that regulated differentiation of various cell types, including the hepatocytes here shown in red, the acinar cells of the pancreas in green, and in red as well are the pancreatic beta cells. And for you, those of you familiar with the mammalian organs, again, you can see a very close resemblance at these early embryonic stages to mammalian development. Of course, one of the keys of forward genetics is to find more or clever and creative screens, if you wish. And so Amnon Schlegel, who is the endocrinology fellow who used to be in the lab, is now at the University of Utah, came in and he was interested in hepatic steatosis. And essentially, he piggybacked on this earlier screen I mentioned and developed a very simple stain using Olredo at day 6 of development, and essentially was looking for mutants where you had abnormal accumulation of neutral lipids in the livers of these animals. And he's now working out the positional cloning of one of these mutants and has uncovered a new player in this, again, very important process. So, as I mentioned, the focus of the talk will be on glucose homeostasis, and thinking about glucose homeostasis in very simple terms, and thinking about the organs involved in glucose homeostasis. Of course, the liver makes glucose, the muscle and the brain take up glucose, and the endocrine cells in the pancreas regulate the synthesis of glucose in various ways. And so in thinking about regulating or coming back, restoring glucose homeostasis, one of the major approaches, of course, has been to increase beta cell mass. Another could be to upregulate the uptake of glucose by muscle or other organs like the brain. And then, of course, a third approach could be to downregulate the synthesis of glucose itself in the liver. And so I'll be initially focusing on the attempts of our group and the field to increase beta cell mass. And then I'll finish off with a new screen where essentially we've designed transgenic zebrafish to look for small molecules that can modulate gluconeogenesis, hoping to find small molecules that in fact downregulate glucose synthesis. So again, the pancreas, of course, is composed of many different cell types. I've already mentioned some of them. The most common cell types being the acinar cells. And the cells that we'll be focusing on are, of course, the pancreatic beta cells, those that produce insulin. And diabetes is, of course, a disease of the beta cells, either in type 1 diabetes, where the immune system destroys the beta cells, but type 2 diabetes, of course, can also be seen as a beta cell problem in the sense that there is inadequate levels or signaling from insulin. And so again, there are various ways that have been approached, that are being approached in the field in terms of curing diabetes via increasing beta cell mass. One of the most talked about approach is to use in vitro differentiated beta cells, either from human ES cells or human iPS cells. And as I said, there's a lot of effort in this way, or in this approach. And of course, trying to understand how the embryo, be it the mouse embryo or the fish embryo, goes about doing this is very important in order to optimize the various protocols used to differentiate beta cells in vitro. Another approach would be to take advantage of whatever capacity the pancreas has to regenerate in order to increase beta cells. And I'll say more about this in a minute. And then finally, or a third approach would be to reprogram cells that are abundant, such as the hepatocytes or the acinar cells of the pancreas, and get them to become beta cells, or at least function like beta cells, and thereby increase beta cell mass. So the regenerative capacity of the endocrine pancreas is in controversial area in the sense that there is a group of thought that in fact indicates that there are no stem cells in the adult pancreas, but that all beta cells originate from other beta cells via proliferation. There's another school of thought, and they believe that in fact the intrapancreatic ducts within the pancreas itself contain stem cells that during injury or even during homeostasis can differentiate into beta cells. And so we've been trying to use the zebrafish to address some of these issues, and as I mentioned, one of the advantages of the system is really to get single cell understanding of cell biological processes. And so I'll tell you then a little more about the intrapancreatic ducts and how the differentiation of some cells in the intrapancreatic duct towards the endocrine sulfate is regulated. And of course, the long-term goals here is once we have a good understanding of this process in the fish, then we can also then move into the mouse or the human and see if in fact one can boost the regenerative capacity of the mammalian pancreas, the intrapancreatic duct, towards increasing beta cell mass. much like many and most other organs, the development of the pancreas is conserved between human and fish, and this is just now looking at the morphological level. This organ is formed from multiple buds, but if you look closely at the various signaling pathways or molecular cascades involved in the formation of these organs, again, you see a very high degree of conservation. So, for example, looking at the intrapancreatic duct, and here in light blue, and again, this could be fish or a human, you can see these intrapancreatic duct cells, and a few of these cells delaminate, differentiate into the various endocrine cell types, which then aggregate to form the islet. And so taking a section of a human adult pancreas, for example, this is from the work of Susan Bonoria, you can see again, these are the duct cells right here, and you can see high levels of insulin expression in some of these duct cells. Similarly, if you look in the fish, and of course the scale here is a little different in the sense that this is the entire pancreas of the fish now. This is the main islet. In red, you're seeing the intrapancreatic duct cells, and we're looking at the same larva now at 5 days and 7 days. You can see, in fact, that some of these intrapancreatic duct cells have differentiated to express insulin GFP and maintain the expression of H2BM cherry, hence they appear yellow. So again, very similarly to what happens in human, some of these intrapancreatic duct cells are differentiating into pancreatic beta cells. Now, this transgene here showing driving the DS-RED expression, or the mCherry expression, is in fact driven by a Notch-responsive promoter. And so from doing a number of experiments along the lines or using these and other transgenes, what we found is that we have 3 different populations of cells in the intrapancreatic duct based on the levels of Notch signaling. So cells that express high levels of Notch signaling cycle slowly, so they proliferate slowly, and they are the duct cells. If these cells now lose Notch signaling, they delaminate and differentiate into endocrine cell types and then aggregate to form the islet. And then in order to close the gaps left behind by these delaminating cells, we have a subset of duct cells that express intermediate levels of Notch signaling and then will proliferate. And we've used a number of tools, and again, I mentioned the ease of doing transgenesis in the fish, and this has certainly been key to these studies. So, for example, we've used transgenic lines where we're expressing under this Notch-responsive element a very stable reporter that I showed you before, the mCherry hooked to H2B. We're also expressing a less or unstable reporter, venous pest, and so we've been able then to use the difference in stability of these reporters to essentially look at the dynamics of Notch signaling. We've also used transgenic effector proteins, for example, a dominant negative version of suppressor of hairless to block Notch signaling, or an ICD, intracellular domain version of Notch, to activate Notch signaling. And so again, just to show you a few data slides, this is now the development of the pancreas. Again, we're looking at the entire pancreas right here. This is in blue now, is the islet, where most of the endocrine cells are located. Here is the intrapancreatic duct. And just paying attention to this one single cell here, which is positive for insulin, also positive for H2BM cherry, but negative for the venous pest reporter. So what this means is that this cell stopped or lost Notch signaling and became insulin positive. So it was initially exhibiting high levels of exhibiting Notch signaling, lost Notch signaling in its recent past, and then differentiated into pancreatic beta cells. So this is the kind of resolution, single cell resolution, that one can easily obtain with this system. And similarly, I mentioned the various tools that one can use to manipulate Notch signaling. Much of them have been used here by Ajay Kitnis in this work. But for example, in addition to chemical inhibitors shown here, one can also use dominant negative versions of effectors or activated versions such as the Notch ICD. And then finally, and I mentioned at the beginning, the advantages of using the system to do live imaging. And so what I'll show you here is a brief movie where again you're looking at these intrapancreatic duct cells in red. these cells here in green are early endocrine progenitors that have recently delaminated from the duct, and as they delaminate and leave a gap in the duct, then this cell will divide, as you'll see, and essentially one of the daughter cells will migrate posteriorly to fill this gap. Here, this one shown in white. So again, even in the late stage animal, one can obtain some fairly high resolution live imaging movies in order to gain additional insight into the process. So essentially, and just to summarize quickly what the contributions of the zebrafish field, and now we're talking about only a handful of labs essentially having been involved in this work for the past 5 years, we've done through forward genetics, the field has identified essentially new and critical insights into the role of, for example, retinoic acid, FGF10, PTF1A, a transcription factor, BMP, c-Met, other signaling molecules in critical processes in the differentiation of the pancreas as well as the endocrine progenitors. In terms of stem cell biology and the idea of trying to boost the regenerative capacity of the pancreas, again, as I've just shown you, we can gain single cell resolution in terms of imaging, but also in terms of manipulation. And we and others have gained then insights into the role of notch signaling into intrapancreatic duct biology and endocrine cell differentiation. And again, the goal here is to take these findings to the mammalian model and see if, in fact, just by activating or modulating notch signaling, one might be able to, again, boost the differentiation of intrapancreatic duct cells into beta cells. And then finally, reprogramming, again, the ease and rapidity of transgenesis has allowed the field to essentially test whether upregulating or downregulating factor X or factor Y can lead to, for example, hepatocytes differentiating to beta cells or eosinophils into beta cells. And because of the high conservation of the molecular pathways and the genes involved in differentiating these various cell types, the findings that in zebrafish would be easily translatable in mammals. And just to show you a recent work from our lab, Dan Eselson has been able to essentially convert eosinophils towards endocrine cell differentiation by downregulating the function of PTF1A, a transcription factor involved in the differentiation and maintenance of eosinophils. And of course, this is very much a line or akin to work, for example, going on in Doug Melton's lab, where they've used 3 transcription factors, now positively acting transcription factors in the beta cells to, again, induce conversion of eosinophil to endocrine cell fates. So this is the level of using the simple or biological tools, forward genetics, imaging, transgenesis, to look at some of the biology underlying critical processes in pancreas development and beta cell differentiation. And as I mentioned, what I really want to spend time doing is another use of the zebrafish that takes advantage of carrying out these large-scale screens. And again, if you think about chemical genetic approach to regulating glucose homeostasis, one can think of several different screens. So you could think about screening for small molecules that increase beta cell mass, or upregulate glucose uptake, or modulate and downregulate gluconeogenesis. So again, one can think about upregulating or increasing beta cell mass, for example, by finding small molecule enhancers of beta cell regeneration, or enhancers of beta cell proliferation or neogenesis, or conversion in the context of reprogramming, or again, as I mentioned, by lowering glucose levels. So I'll focus first on trying to find small molecule enhancers of beta cell regeneration. And the punchline of this work is that after screening 7,000 small molecules, we've identified a signaling pathway that appears to be critical for enhancing beta cell regeneration via beta cell proliferation. And I should mention at this point that, in fact, a parallel, although completely independent screen using rat beta cells in Doug Melton's lab has converged on the exact same signaling pathway. So validating, again, further validating the use of the fish system to identify molecules important and relevant for mammalian biology. So small molecule screens in zebrafish is not a new concept. In fact, many groups around the world have been using the attributes of the fish to look for small molecules involved in their process of interest. And of course, one can think of many reasons, including to identify entry points into biological mechanisms, much like mutations can provide entry points. And then, of course, the other goal would be to find new targets or even possibly new therapies in the process of interest. So we've been carrying out some of these screens for several years now, the first one especially, and then we've more recently started essentially devising new screens to take advantage of new technologies to look at beta cell proliferation, for example. Now, and I'll start with beta cell regeneration. Of course, in order to look at regeneration, one of the first goal of our work was to develop a cell ablation system, and specifically an ablation system that could lead to the elimination of the beta cells at will. And so we turned to, now several years ago, we and others published this model of the system whereby we use a bacterial enzyme called nitroreductase that essentially converts a non-toxic prodrug, this antibiotic metronidazole, into a toxic compound, which if the system works well, and I'll show you that it does, in fact, then essentially will lead to the death of the cells expressing the enzyme, but not the neighboring cells. And we tested this system in 3 different cell types: the myocardial cells of the heart, the pancreatic beta cells, and the hepatocytes. And the system works well in all systems, and of course I'll focus on the pancreatic beta cells. And again, show you using a number of different transgenic combinations. So for example, here we're looking in red at insulin expression, the DS-RED driven by the insulin promoter. And you can see that after exposure, and this is now within 24 hours of exposure to this prodrug, you can see that most of the pancreatic beta cells are eliminated, whereas the surrounding alpha cells, which form a shell of the islet, remain spared. And this is just to show you another set of transgenics, or another transgenic combination that I showed you earlier. And again, here is the islet with the pancreatic beta cells, which after exposure to the prodrug are eliminated. Importantly, of course, if you remove the prodrug, these beta cells will regenerate. So this is before, after ablation, and then 48 hours after washing out the prodrug. And so essentially we use the system to carry out a small molecule screen, you're putting 3 zebrafish larvae per well, and then screening in 96 well plate collections of chemical libraries. And at the time of ablation, just to give you a better sense of the system, there are about 35 beta cells that are mostly killed after 24 hours of exposure to the prodrug. And then essentially after 48 hours of regeneration, the control larvae have between 2 and 5 beta cells. And so these then, we essentially take these animals under an inverted microscope to count the number of beta cells. And I'll only tell you about some small molecules that essentially doubled the number of beta cells. So these are some of the chemical libraries that were screened. This is now a mix of libraries, for example, the sigma-low-pack libraries, which are mainly composed of FDA-approved compounds with known mechanisms of action. And there's also, for example, the Vanderbilt library, which is mainly compounds with no known mechanisms of action. And after screening these and other libraries, we essentially identified 5 compounds that led to a doubling of the number of beta cells after regeneration. This one, as I mentioned, which is coming from the Vanderbilt library, doesn't have any known targets, and so we are working actively to try and identify not only a mechanism of action but also a target, whereas these 4 compounds now, in fact, all converge on one signaling pathway. And just to show you and give you some numbers and show you and tell you more about these compounds, NECA, which is the most potent compound in our assay, is an agonist for adenosine receptor. This compound inhibits adenosine kinase, and I'll show you the pathway in the next slide so that you can get a better sense of where these various molecules affect the pathway. And then these 2 molecules inhibit phosphodiesterase 3 and 4. And again, just to show you compared to the control, this is the number of beta cells that are seen in the regenerating larvae after exposure to these various small molecules. And you can see clearly a large number, or much larger number of beta cells here in this treated embryo than in the control. So adenosine signaling is shown on this slide, and essentially, again, just to orient you as far as the small molecules that were identified, this small molecule blocks or is an inhibitor of adenosine kinase. NECA is an agonist, non-selective agonist for adenosine receptor. We have 2 inhibitors of phosphodiesterase. And so when we identified these compounds, then we went back to the chemical libraries to look at additional modulators of adenosine signaling and screen them at higher concentration, and in fact identified additional small molecules. So for example, this small molecule is another agonist of adenosine signaling, or the adenosine receptor, and this one inhibits adenosine deaminase. And so essentially the activity of these compounds lead to a model whereby either increasing the levels of extracellular adenosine or directly activating the adenosine receptor leads to the effect that's seen as far as beta cell regeneration. So adenosine in the pancreas in fact comes from, or is along ATP release, which is released from beta cells along with insulin. But also, and in the context of our model of ablation, in fact, ATP is also released from cells that undergo injury. So it's very possible that, in fact, in our injury model, after ablating the beta cells with nitroreductase and MTZ, that in fact there's higher levels of ATP release leading to higher levels of adenosine. Yet we can stimulate essentially and lead to higher regeneration of beta cells by overstimulating this pathway. So it was, of course, important to try and understand how the stimulation of the adenosine signaling pathway was in fact leading to this increase in the number of beta cells 2 days after washing out the prodrug. And so it was formally possible, and in fact the stimulation of adenosine signaling was leading to this increased survival of the cells that were targeted by nitroreductase. And so in order to test this formally, we used a photoconvertible fluorescent protein called KD to essentially then label all the beta cells that were differentiated by 3 days. So converting now from green to red, and then look after ablation and regeneration to see how many cells in fact were expressing or were showing both green and red fluorescence. And so, for example, here you're looking at a control animal, and we in fact find very few, in this case 2 cells, that have survived the ablation, if you wish. And then after NECA, for example, in this animal, there are no such double positive cells. And these observations are quantified here. So throughout, either in the control or the experimental animals, you can see low number of cells that survive this ablation. And you can see the regeneration in the control, and again, the much higher regeneration in the experimental animals. So it appears then it's not survival that's increased, if you wish, via the stimulation of the adenosine signaling pathway, but in fact the regeneration of the beta cells themselves. And I'll come back to, or come to a minute, into how this regeneration increase might be happening. So before moving on, we of course wanted to see if these new beta cells and the increase in beta cells were functional. And so we measured free glucose levels in the animals both during ablation and after regeneration. And these results are shown here. And essentially, again, there are some here controls as far as the non-transgenic embryos after treatment with the DMSO NECA. And I'll come back to this in a minute, but essentially we're comparing here the green and the yellow line. The yellow line is now the after ablation and exposure to DMSO, and the green line is after ablation and exposure to NECA. And you can see there's a more rapid restoration to normal glucose levels in the NECA-treated larvae compared to the control animals. So again, suggesting that in fact these new beta cells are functional. So how might beta cell regeneration increase after stimulating adenosine signaling? And again, a number of possible explanations come to mind. One is that beta cell proliferation itself might be stimulated. Another is that there's increased neogenesis or differentiation of the progenitors to the beta cells. And another possible mechanism is that there is transdifferentiation of alpha cells to beta cells. And this is in light of some recent work by Pedro Herrera in Switzerland, where he showed that after ablation of 99% of the beta cells in mouse, most of the new beta cells are coming from alpha cells. And so I'll show you, and I won't show you all the data, but essentially I'll show you some of the data that in fact indicate that stimulation of beta cell proliferation, and specifically newly differentiated beta cells, appears to be the mechanism by which stimulation of adenosine signaling is working. So the fish pancreas, like the mammalian pancreas, is formed from two buds, ventral and dorsal bud. And in fish, dorsal bud contributes exclusively the initial wave of endocrine cells, including the beta cells. So we wanted first to look whether stimulating adenosine signaling had any effect on the number of the dorsal bud-derived beta cells, because we had done some very careful and detailed study looking at the number of beta cells that come from this dorsal bud. And in fact, again, looking and quantitating these results here, you can see that there's no significant increase in the number of beta cells after stimulation of adenosine signaling compared to the control. So it did appear that in fact not all beta cells can be stimulated in terms of their proliferation by adenosine signaling. Looking after ablation now, one can see in fact a very different picture, and it is shown here. So again, both looking at by confocal imaging as well as quantitating, you can see again there's a significant increase in the number of beta cells after stimulation of the adenosine signaling pathway. But you can see that in fact most of these beta cells now have incorporated EDU, and we're doing the EDU treatment from day 4 to day 6. Day 4 is when we wash out the drug, so this is the beginning of the regeneration process. So most of these cells now, as indicated here, or these cells have in fact incorporated EDU in the past 2 days, so they have recently proliferated or undergoing DNA synthesis. And this is a very different picture that you see if in fact you look and do this very similar experiment but without ablation. So you just take control animals and expose them again to DMSO or NECA, and again there's no very significant increase in the number of beta cells. There's a small and significant but minor increase now in the number of beta cells in the NECA treated animals that incorporate EDU. So there's a small effect on proliferation on beta cells in the absence of ablation, but a very strong effect in the presence or after ablation. So again, so what these data indicated then is that the increase in beta cell regeneration is coming from an increase in beta cell proliferation. And so if you think about the differentiation of progenitors to the beta cells, again, it's important to try and understand which cells not only are differentiating to beta cells, but which cells are proliferating. So it could be, again, if you think about this transdifferentiation model where alpha cells are differentiating into beta cells, or if you think about the progenitors, to try and understand at which stage are these cells being essentially affected by adenosine signaling. And again, I won't show you all the data, but suffice it to say that it appears that the newly differentiated beta cells are the ones that are affected by this increase in adenosine signaling. So for example, and I'll just take another endocrine cell type, we'll just look at the alpha cells. And as I mentioned, in the mouse model of beta cell ablation, the alpha cells become transdifferentiate into beta cells. Hence, we wanted to look at the alpha cells themselves, although we've looked at other cell types in the larva. And again, you can see here in the confocal images or in the quantitation that in fact here in green are the alpha cells, in red are the beta cells, and again there is very little incorporation of, or this difference, EDU incorporation after stimulation by NECA compared to the control. But you can see again the beta cells, most of them have incorporated EDU. So the working model then for this data is that NECA expands the beta cell mass by essentially stimulating the proliferation of these newly differentiated beta cells in this ablation/regeneration model. But in the absence of ablation, NECA has a very limited effect. So in fact, there is no proliferation or stimulation of proliferation unless the animal has need for new or more beta cells. So there are, of course, a number of questions that remain to be asked or answered as far as how adenosine signaling itself is regulating the proliferation of the beta cells. One, we'd like to understand if this effect is direct, meaning are these beta cells now directly responding to adenosine signaling, which receptor are they expressing, and so on. As I mentioned before, Doug Melton recently was talking, in fact, at a recent meeting where Francis Collins gave one of the keynote addresses. This is the Beta Cell Biology Consortium, funded by NIDDK. And he showed some data from his lab where they were using a dispersed rat beta cell model and essentially screened small molecules for compounds that simulated the proliferation of rat beta cells and identified an inhibitor of adenosine kinase as a potent stimulator of beta cell proliferation. So here we have two very different screens, one done in the whole organism, the fish, another one done in cell culture, and in fact both converging on the same signaling pathway. So the hope is that in fact the adenosine signaling in the context of beta cell regeneration and increasing beta cell mass is going to receive increased attention as far as now trying to get not only to mechanisms but also to see if it can be validated in other animal models of diabetes and of course in humans. We're also looking at the effect of adenosine signaling in a number of contexts of human biology. So with Al Powers in golden color, we're looking at human ES cells differentiating into beta cells. We're also looking at SNPs associated with adenosine receptors in the context of type 2 diabetes patients. I want to just finish this part by talking about a paper that was in fact published 4 years ago now. And this group was using a couple of mouse models of type 1 diabetes, and in fact published that NECA, this adenosine receptor agonist, can in fact improve glucose homeostasis in these models. As you can see, this is now in the presence of NECA compared to control, and we're looking here at blood glucose levels. But these investigators, in fact, concluded, although they did not look at beta cells at all, that in fact the beneficial effect of NECA in these mouse models of diabetes came from modulating the immune system. And so, of course, now we're going back to these studies and going back to these models and to see if, in fact, whether NECA may, in fact, be playing a role in beta cell proliferation in these models, maybe in addition or only in addition to the immune modulation. So in the last 5 to 10 minutes, what I want to do then is to take, again, as I mentioned, trying to get and look at glucose homeostasis via another angle, another direction, and that is looking at whether we can find, or one can find, small molecules that would reduce the glucose output by the liver. And so, again, just a simple cartoon here showing essentially the input of various hormones from the endocrine cells into the liver in terms of affecting gluconeogenesis. And many of those hormones converge on the activity of an enzyme called PCK, or also known as PepCK. And essentially this enzyme, and this is a complex cartoon here, fits right here in the gluconeogenesis glycolysis pathway, but converts oxaloacetate into phosphoenol pyruvate, is one of the key enzymes and a regulated enzyme in gluconeogenesis. So the idea is then, can we find or can we make a transgenic line where essentially one could examine or identify small molecules that modulate the expression of this enzyme. And this was a project that was carried out or initiated by Philip, another postdoc in the lab, and essentially he identified a promoter element from the PepCK or PCK1 gene in fish, and in this case hooked it up to Venus and looked at the expression pattern. and then went on to essentially, as I'll show you, hook it up to luciferase to try and then get more quantitative readout of the activity of this promoter. But you can see already that there's specific activity with this 2.8 kb element, specific activity in the liver. And if you compare now various feeding regimens, so this is now basal levels, if you now add glucose to the water, which will lead to the secretion of insulin, you can see downregulation of the activity of this promoter. And then you can get further downregulation of this promoter in the presence of glucose and high concentrations of metformin, a type 2 diabetes drug. So again, suggesting that in fact this promoter responds as appropriate or as one would expect from the biology of the system. So as I mentioned, in order to get more quantitative, Philip turned to make now a luciferase line and essentially used a number of small molecules that are known to upregulate PEP-CK promoter activity and to see how this luciferase reporter was behaving in this assay. So what you're looking at here is 3 different drugs that promote the activity of this enzyme, the activity of this promoter for this enzyme in cell culture. And you can see that increasing concentrations of dexamethasone, forskolin, or isoproterenol lead to an upregulation of the activity of this promoter as measured by luciferase activity. Again, to try and get a better sense of the dynamics of this reporter, what Philip did then is essentially maintaining concentration of isoproterenol, a positive regulator of the promoter, and then increasing concentrations of metformin. And again, you can see the activity of this promoter decreases as increasing concentrations of metformin are added to the water. And then again, to see if this increasing activity of the reporter or the promoter would lead to increasing concentration of free glucose, much like I showed you for the pancreas work. You can see again that now increasing concentrations of metformin will lower the concentrations of free glucose, again in agreement with the lower activity of this luciferase reporter. So essentially now the system was ready for a small molecule screen, and one where in fact there was a high and dynamic range of the activity of this reporter and high sensitivity for the screen. And very simply again, and Philip concentrated now on the low-pack library from Sigma, which are mainly FDA-approved small molecules with known mechanisms of action, and using 3 fish per well, and essentially bathing the larvae from 3 days to 5 days, and then doing luciferase assay at the end of this treatment. What he was looking for, as I mentioned earlier, are of course inhibitors of the activity of the PEP-CK promoter, because these might then translate into lower glucose production by the liver. But not surprisingly, from the biology and the long literature that and many years of work by many lab before, what he found is mainly activators of the activity of this promoter. And these are some of the numbers here in terms of the toxicity and the primary hits. But what he found, and what I'll finish off with, is a number or 2 interesting findings. And the first finding is shown here. And essentially what you see here are some of the activators of the activity of this promoter. And so again, some of them were well known, beta-2 agonists, glucocorticoids, but there was also now a number of antipsychotics and antidepressants shown here. And those are in fact very potent activators of the PEP-CK promoter. In fact, these drugs, these FDA-approved drugs, had in fact been associated before with the high risk of diabetes. But in fact, this is one potential mechanism, in fact, that this could be happening, that these drugs are directly affecting the promoter of PEP-CK, increasing the activity of this promoter, increasing gluconeogenesis, thereby increasing glucose levels. The other finding, and that one that's more interesting to us, is that he identified 2 drugs, 2 small molecules that are shown here, and these small molecules increase the activity of the promoter but lead to a lower free glucose levels. And our current interpretation of the activity of these drugs is they in fact induce a fasting response. And so they, in fact, induced an increased uptake of glucose by a number of peripheral tissues, which would lead to lower glucose levels, which would then in turn lead to an increase in the activity of PepCK. And more recently, Philip has looked at the activity of this one compound in mouse, and in fact now has got some very encouraging results that in fact this compound could be inducing a fasting response in mammals as well. So then to summarize these chemical screens, what I've shown you is that, of course, the zebrafish, and as I mentioned, this is now being used by many people in the field. There's some examples on hematopoietic stem cells by Len Zahn. There's work also by Randy Peterson, both in Boston, in terms of using the system to try and identify either new targets or new therapies to approach diabetes. What I've told you is that, in fact, as far as beta cell regeneration is concerned, that we've developed a model where we can efficiently kill the pancreatic beta cells, that the small molecule screen has identified the role of adenosine signaling in increasing beta cell regeneration via increasing beta cell proliferation. I told you about gluconeogenesis and the screen for small molecules that regulate the activity of the PepCK or PCK1 promoter, and in fact that two of these compounds that were identified in the screen may be inducing a fasting response, which hopefully will then be translated or will translate into a similar response in mammals and can be then further studied. And then I won't have time to show you, but we're using a new reporter system called the Fuji system developed in Japan to look at proliferation of beta cells in a live animal, and again looking for small molecules that will enhance beta cell proliferation. So I just want to thank the people who have been driving these projects. I mentioned them along the way. Nikolai has been involved in the intrapancreatic duct work, Olaf and Philip on the chemical screens that I mentioned. Also a number of collaborators at UCSF and other places, and support from NIDDK, the BCBC and JDRF for the work I presented today. And I also want to end by again thanking Dr. David and Arias for the invitation, and some key people at the NIH who've been helping with extramural funding. So I'll thank you for your attention, and be happy to take questions.
Sushil Rane, the NIDDK, very nice talk. So several years ago, we published on cell cycle regulators in beta cell proliferation using mouse systems, and stemming from that work, we realized that there is an age-dependent regulation of replication. So in this model, what's the natural kinetic of beta cell proliferation as far as replication is concerned? Right,
that's a very important point. So we're now using this Fuji system that I just mentioned to get a better sense of proliferation. We have seen proliferation in the adult, but we haven't been able to quantitate it to the level of rigor that we'd like. So, and again, it depends, very much on the feeding regimen of these fish and so on. But that's an important question, and certainly, we also want to move as far as this regeneration model away from the larva into the adult. One key question, of course, is how much work to do in the fish before you move into the mouse. And certainly, I think in terms of the adenosine signaling pathway, especially with the results from the rat beta cells and from Doug Melton, I think now all indications are that one should move into mouse, at least in terms of looking at efficacy. One could still use the fish to try and dissect mechanisms, but I think mammalian studies are certainly warranted.
One more follow-up, if I can. So as far as any mutants that you could find which basically are limiting the beta cell proliferation ability, means any pathways, adenosine obviously is pro-proliferative. Any on the other side which might be growth cessation pathways?
Right, so for that, we have not found any small molecules that essentially will lead to a block of beta cell regeneration in this model. And again, that's why we're moving now to this beta cell proliferation assay using the Fuji system, right, which has this two-color system that allows you to differentiate between cycling and resting cells. And there we might be able to identify small molecules that essentially prolong the resting period. I think this would be a more sensitive assay.
Thank you, super.
Thank you.
Olivier Blondel, NIDDK. So with regard to the adenosine signaling pathway and with sort of an eye on translation, do you expect that you will need some sort of pancreas specificity for activity? And if so, are there elements of this pathway that are specific to beta cell?
Right, so Olivier is raising a very important point. So I didn't show you the data, but we looked at other endocrine cell types, we looked at acinar cells, we looked at hepatocytes, and we're not seeing any effect on the proliferation of these cells after essentially adding or stimulating the adenosine signaling pathway. Clearly, these receptors are present in many different tissues, but for some reason, only the beta cells and only the beta cells in animals that are in need of additional beta cells are responding in terms of increasing their beta cell proliferation. So, there are some critical biological issues, some mechanistic issues that we need to understand there, but certainly one doesn't need to think about the need to find a beta cell specific receptor, if you wish, to affect beta cells specifically in this model.
Congratulations for looking at the issues of diabetes from the zebrafish. Now, one of the issues that come up that a lot of the lower level mammalian systems or non-mammalian systems have this unique ability for regeneration, but somehow that ability has been lost. So things what we are learning in the lower vertebrates, will it be really meaningful in mammals like us, or what are the issues that might be helping us to understand what we lost, or we might be trying to get it back?
Right, so I think much like people talk about no turnover of cells in the heart, in the mammalian heart, and no turnover of beta cells. And I think these are extreme statements that, at least in the context of the heart, have been recently shown to be wrong. So I think mammalian and human organs do have some limited capacity to regenerate and to turn over their cells. And certainly not as extreme as what the fish exhibits, but there is a basal activity, and what our hope is that one can boost that basal activity. And certainly by looking at the mechanisms that happen in the fish, one could then dissect this path towards proliferation or regeneration and try to understand which step is in fact limiting in the mammalian system. So what's become clear, for example, in the case of heart regeneration, is that the way the fish heart regenerates is by de-differentiating mature cardiomyocytes, which will then proliferate and redifferentiate. So now I think the mammalian fields can learn from that and say, well, how can we make mature cardiomyocytes de-differentiate? Because maybe that's half the problem, right? So, and again, very similarly, in the pancreas field, if we can understand how a lowly organism like the fish does it, then I think we can ask more precise questions about why the mammalian pancreas or beta cells do not do it.
So I had a question that I was just thinking about. So it almost looks like the adenosine pathway, or using NACA, affects regenerating cells rather than necessarily beta cells. So what about regenerating liver cells? Are they affected?
Right, so that's a very good question. So as I mentioned or showed you, we have generated this ablation model for myocardial cells as well as hepatocytes. And Donghan Shen, who recently moved to Pittsburgh, set up his lab, recently developed a stronger hepatocyte-specific nitroreductase line where you can get a more robust ablation. And we've sent him our top 20 hits, small molecules that affect beta cell regeneration, and none of them appear to be affecting hepatocyte regeneration. So this appears to be quite specific to beta cells. Of course, now he's going in his lab then to try and do a small molecule screen to look for enhancers of hepatocyte regeneration, and of course we'll be interested in testing his compounds.
Well, if there are no further questions, then I'd like to thank Didier Stenier for a wonderful lecture and invite everyone to a reception in the library. Please come join us there. Thank you.
Thank you.